Today, Xiao-Wei will talk about his fantastic work on an interesting aspect of COP2 code that plays critical role in lipo-homeostasis. Without further ado, let's welcome Xiao-Wei.
Well, first I want to start by thanking Yihong for organizing this online webinar. And it's really a distinct honor for me to be the first speaker of this year, the inaugural speaker. And also want to thank Yusong for the very generous introduction. And to all of you, wherever we are, I hope it's not too late to wish you a wonderful year of 2024. So as Yusong has mentioned that, I think my research program really aims to take the very basic cell biology to physiology and even to potential therapies for major human diseases. So for that, then I will start by introducing an experiment that we are very excited about, the reversal of deadly atherosclerotic plaques in live mice. And this is, I should say that this is a goal that has been long sought for in clinical settings. However, this is not even well achieved, even in the preclinical mouse models. And we were able to devise an approach in these murine models. You will see that that's entirely originated from our basic research and achieved the reversal and even to some extent almost the elimination of these deadly atherosclerotic plaques in the artery regions in the vessels in these live mice. And we're certainly very excited about these results because it's needless for me to say that cardiovascular diseases had continued to be the #1 killer worldwide. And it really claims more than, I think more than 1 out of every 3 human lives that were lost. However, I do want to mention that dyslipidemia or elevated levels of lipids in the circulation is the most prevalent, most common cause for the top killer cardiovascular diseases. And the elevated lipids in the blood, in the circulation would silently build up these deadly atherosclerotic plaques, and multiple epidemiology studies have shown that more than or about at least about half of the adult population have blood lipid problems to some degree. So lipids in the blood is really a major medical problem. On the other hand, lipids, due to their very unique hydrophobic nature, they have to be packaged into these lipoproteins, which are actually secreted into the bloodstream so they can be taken up and utilized by the various different cell types in these different extrahepatic tissues. And the lipoproteins as shown here, in terms of secretion, they really represent a very unique class of secreted cargoes. For example, they're huge in size and enormous in number, and their biochemical property are entirely different from the general secreted proteins that we tend to think of, such as insulin or even the very abundant albumin. And in my opinion, how would these lipid cargoes, very unique and very important, travel through the numerous lipid membranes in the cell still remains quite a mystery. And that, as far as I can tell, has actually attracted some of the most brilliant minds in science. So one of the, I think one of the most important advances in this very common problem of lipid regulation, lipoprotein transport, has to do with the identification, the discovery of the low density lipoprotein receptor or the LDL receptor. And this discovery was made by really the 2 giants in the field, Drs. Michael Bowen and Joe Goldstein. So the function of the LDL receptor is to localize to the cell surface, especially liver cells, where they mediate the specific recognition of the lipoproteins in the circulation, in the blood, and then enable their subsequent uptake and clearance from the circulation. So, and the discovery of the LDL receptor really has tremendously advanced both basic science as well as translation medicine. The receptor is actually the ultimate target of the most widely prescribed lipid-lowering drug, the statins, which will have to elevate the level of the LDL receptor to achieve lipid lowering and therefore cardiac protection. So about 10 years ago, Yusong and I actually started a lab at about the same time. When I started to set up the lab, I was very much intrigued by this classic landmark discoveries in the receptor-mediated lipoprotein uptake. So I then actually choose to study the converse or even the more upstream process. How do these lipids get into the circulation at the first place? And for that process, that was indeed entirely inspired by the landmark discovery of the LDL receptor. So we had for lipid secretion into the circulation, we had a very simple hypothesis, and that is if the lipoproteins in the circulation needs a specific receptor, the endocytic LDL receptor, to get into the cells, then would it be possible there will be a different secretion receptor for these lipoproteins to get out of the cell? So following this hypothesis, we really combine the power of rare genetic disease, and also population genetics in humans. And that led us to a ER transmembrane protein by the name of SIRT4. Some of you are quite familiar with this. And we further show that SIRT4, this transmembrane protein at the ER, function as a transmembrane bridge that connects these unique and important lipoproteins to the cellular transport machinery, the COPII complex. Therefore, it potently regulates lipoprotein secretion and therefore lipid homeostasis in both disease and in the health and disease states. And we start to publish in this area a few years ago. And for this study, perhaps the most striking piece of data in this study can be summarized in this panel shown here. These are actually experiments performed in the LDL receptor knockout mice background. And when mice are missing their LDL receptor, they fail to clear lipids from the circulation. And if you still feed them with Western diet, their plasma lipid level will shoot up to over 1,000. And that's also 1,000% higher than the normal 100-ish range that you're observing in wild-type mice. And in these completely pathological dyslipidemia mice, we just need to inactivate one single gene, SIRT4, and the inactivation only needs to take place in the liver, and that's sufficient to deplete the skyrocketed plasma lipid levels to near zero. And as a result, these double mutant mice are completely protected from cardiovascular complications such as atherosclerosis. So I think, to my knowledge, I think this will hold true. And this is the most profound lipid lowering effect from a single gene knockout in a live mice, because in a live mice, one would not be able to observe plasma lipid levels in negative values. And we further find that this very profound lipid lowering effect by SIRT4 knockout is also dosage dependent, in that we just need to partially inactivate the SIRT4 receptor, and that would lead to a correspondent lowering of plasma lipids as well as cardiac protection. And this dosage effect of SIRT4 is actually very much consistent with the population genetic hints that led us to this important regulation important regulator of lipid homeostasis. And this part was actually a collaboration initially with Kristen Willer when she was still at Michigan. Now she heads the the human genetics department at Regenera, and it was completed with her trainee, Professor Xiangfeng Lu, who's now based in China. And this human genetic link will also implicate certainly, and plus the dosage effect, some translation potential of this receptor, as well as the pathways was defined by the receptor. And we also now know that SIRF4 actually belongs to, and perhaps even help assemble a functional receptor complex, which contains a number of functionally very important and mechanistically still remain to be defined transmembrane proteins. And one of these transmembrane proteins by the name of TMM41B actually encodes a long-sought phospholipid scramblase localized at the endoplasmic reticulum. In the process we are studying, TMM41B is responsible for the transmembrane shuttering of the newly synthesized phospholipids located at the cytosolic leaflet into the ER lumen to coat the surface of these lipoproteins that are produced within the ER lumen. So, and that's actually a very interesting, exciting line of research in the lab that I won't be able to cover today. But I think this really showcases, this really demonstrates the opportunities opened up by SIRF4. And the reason is actually very simple, because the production of lipoprotein is actually very complex, involves many fundamental and poorly understood reactions. The production actually is starts within the ER and still continues in the Golgi apparatus, even though these two organelles are very different. And many of the enzymes, regulators will need to write with lipoprotein through Golgi. And likely now we seem to find that they tend to write together with this very important secretion receptor SIRF4. And so for today's talk, I would actually highlight another really unexpected story that was also enabled by SIRF4. So I think work from us, and especially by many others in the field, have long demonstrated that the secretion of lipids in the form of lipoproteins is both highly specific and very, very robust. Now we know that the specificity actually can be defined from very early on, at the very beginning of the secretory journey, and that is upon the exiting of the endoplasmic reticulum. That in large is defined by the SIRF4 receptor, and the simplified or the core mechanism can be summarized here. Basically, SIRF4 will link the unique, very important lipoproteins to the general cellular transport machinery, the COPII complex, many of you are very familiar with. And sitting at the very top of this program is a signaling molecule, a small GDPA SOL1B. And this encoding gene is also mutated in the rare genetic disease by the name of chylomicron retention disease. And that is actually the rare disease link that led us to this whole program. So, but the question to us is, besides this specificity mediated by, defined by the receptor, how is the efficiency of the entire program ensured? Or in other words, how would the very general transport machinery, the COPII complex, accommodate the massive amount of lipids that ride on the numerous lipoproteins? And for that, I would need to highlight two people in the lab, especially Xiao Wang. She's actually the first PhD produced in my group. And I think her intelligence really made a decisive role, as you will see, for us to demonstrate that the condensation of the COPII machinery play an instrumental role in ensuring the whole efficiency of lipoprotein secretion and therefore lipid homeostasis. So, and I think for this well-informed audience, I really don't need to introduce too much about the idea of biomolecular condensation or phase separation, other than saying that this really has emerged over the last 10 years or so as a key mechanism in cellular organization, especially at the mesoscale in these so-called membraneless assemblies, membraneless organelles or condensates. And these oftentimes were driven by weak multivalent interactions through the intrinsic disorganized region that are widely present in many proteins, but really has escaped the investigation of conventional biochemistry and cell biology over the years. Nevertheless, I do want to mention that despite the rapid and very exciting progress of the field of condensation or phase separation, the whole physiology of this phase separation concept is really lagging behind, let alone translating some of the very exciting mechanisms into potential therapeutic approaches for major diseases. And I do hope the following study of ours may be able to bridge some of these gaps. Well, anyway, so the COP2 complex, COP2 phase separation, this is the core components of the COP2 machinery, as some of us are very familiar with, has two layers. The inner layer that direct the C's cargoes as colored in green, and the outer layer here, outer coats colored in red. And we and others have long noticed that the COP2 components in cells can form these very discrete and very enriching concentrated punctures in the cell. And this is especially true when the lipoprotein trafficking, lipoprotein transport is induced in these cells. And in vitro, the inner coats, the inner code subunit of the COP2 machinery, Sec23 and Sec24, when they were purified, they were able to phase separate from the aqueous solution and form these so-called protein or liquid protein droplets, which are highly enriched with the purified recombinant COP2 inner coats. So, and this is entirely dependent on a evolutionary conserved intrinsically disorganized region located at the very end terminals of the Sec24, the cargo selecting subunit in the COP2 machinery. This is well separated from the structural regions of Sec24, so indicating that this can be an alternative complementing mechanism regulating COP2 function. So these nowadays almost become the norm of the condensation or the phase separation field. However, as far as we can tell, the COP2 condensation or the phase separation of the COP2 machinery is very unique in its self-constrained nature. So here, shown here again, so the inner code subunits are colored in green in conditions when the inner codes can phase separate and form these liquid droplets in vitro. The outer code shown here in red actually cannot. Nevertheless, they can actually be recruited by the inner code droplet and then get enriched. So in other words, COP2 condensation can actually function as a assembly promoting mechanism. Moreover, the recruited outer codes actually forms a shell on the surface of the initial inner code droplet and prevent its overgrowth or overprogression into a gel-like state. So this, in other words, this constraint actually provide a balancing mechanism to prevent, to ensure the overall dynamics. And that this is actually very important for the trafficking flow. So we actually also, besides these in vitro reconstitution experiments, we actually also made chimeric mutants and so on and so forth, did all those things and used all those mutants in cells deletion. And we even talked to the physics colleagues in our campus who told us that perhaps the easiest model we can build from all these cellular and in vitro reconstitution experiment is this bell-shaped curve or bell-shaped regulation, which means that when every activity is low, then the concentration would enrich these subunits to promote assembly. And this will proceed to a so-called optimal state or a sweet spot featured by balanced assembly and optimal dynamics. However, passing this optimal state, the overcondensation would actually halt, would damage or will slow down the dynamics or the kinetics, and this would actually decrease the overall output. And another way to understand this, as I was thinking about, is perhaps a real life example where, let's say if I want to take a taxi after this late night talk to go home, and I will maybe call a taxi with Uber or the Chinese equivalent Didi. And if there's no car or no taxi around me, this is no good because I won't be able to go anywhere. We all want the so-called maybe the middle scenario. There's a right amount of taxis that are enriched around us. However, in some cases, and this is actually not uncommon in Beijing, if you have too many taxis around you that already produce a trafficking jam, then this is also no good. I may be able to get into one of the taxis, but still I'm not going anywhere. So consistent with these analogies, when we delete the IDRs of section 24 to prevent condensation or replacing the endogenous one with a IDR that's prone to overprogression, overcondensation. In both cases, these mutants would disrupt lipoprotein trafficking. So, however, we do know that this is not enough. We know that we need to find a regulation to demonstrate the physiological relevance for all these mechanisms, all this modeling. And the regulation actually came from a very unexpected angle of viral infection. And this took us many years, 5+ years to really just to think of what experiment to do to make sense of this. And I hope I can explain to you in maybe 2 or 3 minutes in the next slide. So this is one of the first set of experiments performed in my own lab. So when we decide to study lipid metabolism, lipoprotein secretion, we made a murine model of the human chylomicron retention disease gene SORN1B. So when we delete SORN1B, even only in the liver, this will compromise the downstream COP2 complex in a selective way. And this will block lipoprotein secretion. So in the end, the liver's knockout mice of SORN1B will have lower plasma lipid levels. And these are all very expected, very consistent with the functions of, the known functions of these genes, also very consistent with the human disease phenotype. However, the surprise came when these SORN1B mutant mice, are exposed to adenovirus, which is a very common pathogen in both mice and human. It's so common that we even use the virus, the recombinant ones, for gene delivery. However, when the mutant mice were exposed to adenovirus, the viral infection would actually rescue the diminished lipid levels in the mutant mice. These results were very puzzling to us because it's well known in the field, and we know this as well. Adenovirus in white-type mice, especially at slightly high doses, these would impair lipoprotein secretion and give you reduced lipid levels in the white-type mice. And these are actually completely opposite from what we observe in the SORN1B mutant mice. And this was termed as, in white-type mice, this was termed as adenoviral toxicity. Was not, everybody knows about this, but nobody really paid much attention. However, we do know that these this viral effect on plasma lipids are specific for at least 2 reasons. Firstly, in the surface knockout mice, when we delete the secretion receptor for lipoproteins, the adenoviral infection does not affect plasma lipid levels. In all these cases, viral infection had no effect on the general secreted proteins, on the plasma level of these general secreted proteins, even the very abundant albumin. So from a genetic point of view, these results are not very hard to interpret. This means that the virus must be hitting something downstream of SORB, but upstream of SIRT4, and that would be the COP2 machinery. However, the puzzle for us was that how could the same virus, same adenovirus, produce these entirely opposite effects in these two scenarios? And we had these results for many years, for 5-6 years. We could never think of any possible explanation until one day, Xiao, she was still a student in the lab, came to me and said that, would it be possible if the virus is affecting the balanced, the constrained COP2 condensation that we independently observe in the lab? And it took me an entire day to think this through. And if you already figured this out, that's great. That means you're much smarter than all of us, than at least than me. So what she meant, or what I understood, was that in the SORB mutant mice, when the COP2 complex were compromised, then if you promote condensation of the COP2 machinery, this will rescue the COP2 function and rescue lipoprotein secretion, give you higher plasma lipid level. In the wild-type mice, where everything's already at the optimal state, if you still promote condensation, and this will lead to an over-progression of condensation, and these will impair the whole output and give you lower plasma lipids. And very importantly, in the SIRT4 knockout mice, when you're missing the transmembrane bridge between the lipoproteins and the trafficking machinery, what happens on the COP2 machinery is irrelevant. So once we figured this out, we went on to test whether indeed the viral infection would affect, and in this case, it would be very simple, would promote the condensation of the COP2 machinery. Indeed, in hepatocytes where we used to study lipoprotein secretion, we find that adenoviral infection very nicely enlarge these very concentrated puncta structure of the COP2 complex. We later determined that this is a result of condensation. I will show you evidence in later slides. And even more importantly, because condensation or phase separation is a biochemical reaction, and we then went on test in a pure in vitro setting by mixing, supply the purified inner coat complex of COP2, secondary 3 and secondary 4, with liver cytosol that was extracted from either control mice or mice that have been infected with adenovirus. And we find that in this pure in vitro setting, the adenoviral infected liver lysate was able to promote this in vitro reconstituted COP2 phase separation in a test tube. And here actually came to, I would consider my maybe only intellectual input in this whole project as a PI. So I told Sean, my student, that if the virus is promoting phase separation or condensation of this essential COP2 machinery in such a matter in a pure in vitro setting, it must be through a mediator. And you already have an in vitro assay, so let's go purify it. So normally the students, I didn't expect her to listen to this, but so I was very appreciative. She actually followed my suggestion, even though this could be a crazily risky idea. And she even convinced a junior student to work with her. And then we set out to purify what we think will be a COP2 condensation promoting factor that are present in the adenovirus infected liver cytosol. And luckily for us, we eventually determined that this factor is not a nucleotide because nuclease is digested, did nothing to the activity. It's not even a protein because proteinase K digested did not kill the activity. And the promoting factor appears to be very heat stable, suggesting that this may be a small molecule. And this now becomes very interesting. So we further fraction these based on molecular weight. And indeed, the promoting activity is almost entirely retained in these small molecule fractions. And very serendipitously, this turned out to be very useful. Addition of EDTA, very common chelator in this fraction, in this small molecule fraction, completely abolished the promoting effect. And this will suggest to us the involvement, the contribution of some divalent metal ions. And this may turn out to be very useful. So we then turn to a chemical approach, basically use ICP-MS to screen for the metal ions that was affected by adenoviral infection in immune mice. And in this screen, manganese actually stood out in this screening. So before we get excited about this, though, we do want to ask, is manganese, no, this was very poorly studied, is manganese the promoting factor that we were looking for? We thought so because, again, in this pure in vitro setting with purified recombinant COP2 inner coats, when they were supplied with increasing dose of manganese ions, and it very potently promote the in vitro reconstitution, in vitro condensation of the purified COP2 coats. And this promoting factor is also very specific because none of the other divalent ions we tested, including calcium, magnesium, iron, copper, zinc, none of them had the same promoting factor as manganese. And it turned out, and this was very interesting to us, that manganese can directly bind to the inner coat of the very general, very conserved COP2 machinery, as determined by both either by ITC or IMAC proton with immobilized manganese ion. And this was very surprising to us because manganese was mostly known as, as required but really passive facilitating cofactors for metabolic enzymes. The COP2 complex is not an enzyme. And we later find that manganese actually have a much more active a signaling role in regulating, in controlling the COP2 machinery. And indeed, consistent with this idea that we see that in cells, manganese treatment would very nicely enlarge these puncta structure of the COP2 COP2 machinery as visualized by the Sec24 subunit. And this is actually a result of condensation because when we remove the intrinsically disorganized region of Sec24, the protein will become completely diffusive, completely, with no discrete structure, and manganese treatment had no effect. On the other hand, if we replace the endogenous intrinsic disorganized region with those from the protein fuzz, which is prone to overcondensation. So the chimera protein would have enlarged condensates to start with, and now manganese treatment would easily turn this into a gel state with dramatically halted, with dramatically slowed down kinetics. So basically, manganese can shift the balance of COP2 condensation in cells. And as a signal, we can expect that we may be able to turn the strength of the this type of signal to produce biologic effects. And indeed, when we expose cells to increasing dose of manganese ions, and it very nicely leads to a corresponding promotion of the cellular condensation of the COP2 components. And on the other hand, this would also, in a dose-dependent manner, by promoting condensation to help assembly, this will also gradually decrease the overall kinetics of these structure, perhaps likely due to a crowding effect. And the combination or the balance of these two different, if not opposite effects, would produce would produce the maximum efficiency of this condensation-based mechanism for the COPII machinery. And this will lead to a selective regulation in the secretion of these lipoproteins, but not those general secreted proteins, even the very abundant albumin. And this regulation of lipoproteins also fit with in a bell shape in a bell shape with low dose of manganese being promoting to a peak value, and higher doses beyond this will appear inhibitory. And this actually is quite consistent with this bell-shaped curve that we modeled based on the in vitro reconstitution and chimeric mutants. But the beauty of this is that we can actually we can turn this whole process with just one single variable, and that is the concentration or the strength of the manganese signal. And we further show that actually the hepatic mitochondria can work as the manganese store to supply this messenger to direct the signal to the COPII machinery on the ER surface, and this will lead to a selective and highly efficient secretion transport of the lipoproteins. So basically, then really the question now becomes to us is that, can we harness some of these interesting and perhaps novel mechanisms to physiology, and or even to something that's relevant for major diseases such as lipid disorder, or even the very prevalent cardiovascular diseases. And I should mention that this has been previously known as being well documented in the field that manganese, when supplied with diet, they're known to get enriched very nicely in the liver by the hepatic mitochondria. And these are actually the center of actions for the mechanism that we describe, as summarized here. So we then actually took a leap of faith and basically ask a very daring question. Could targeting COP2 condensation with the manganese signal really controls a major physiological parameter, such as circulating lipid levels? And in other words, we basically are asking, could the manganese regulation enable this very important switch from a quantitative regulation into a qualitative change? And for that, when we take anything that's so basic at the cellular level to in vivo, we know that we need to do a lot of calibration, a lot of control. So we first start by a set of calibration experiments. And basically, this is to validate this dietary regulation approach, to control manganese signal in vivo models. So we basically put mice on a manganese deficient diet and then supply different doses through drink. And interestingly enough, in the Saw and B knockout mice, where COP2 activity is level, the increasing manganese supply with diet or with drink can lead to a dose-dependent rescue or elevation of the diminished lipid levels in these mutant mice. And this effect is actually, we think it's very much dependent on COP2-mediated transport, because in the SIRF4 knockout, in the receptor knockout, even the highest doses of manganese signal was not able to affect lipid levels. So these results would basically show that the dietary approach to elevate manganese signal is effective, and it's also quantitative, and it's also very important and quite selective. So the calibration experiment, can be summarized here, and this really allowed us to ask, or intrigued us to ask, what would happen in normal mice, or at least in mice with normal COP2 function. And the results to us actually, this is greatly to our great satisfaction, also demonstrate, or at least suggest, that the COP2-mediated transport is a major determinant of lipid control in vivo, because we observed that this bell-shaped regulation on plasma lipid levels by manganese signal, and therefore COP2 condensation is maintained in vivo in live mice, even in hyperlipidemia cases as induced here. So basically with low dose of manganese signal promoting to a peak value, and beyond that, higher doses will become inhibitory. And interestingly enough, the peak of this regulation, the manganese dose we use is actually quite similar to the endogenous hepatic manganese concentration that we'll observe in normal mice. This would actually suggest that there's some kind of cooperation or even evolution adaptation for maximum lipid supply in mice. This will also suggest to us that we may be able to purposely elevate the strength of manganese signal by very simply dietary supply to achieve sustained and intensive lipid lowering in mice, in normal, so-called normal mice with normal COP2 function. And this actually becomes very interesting to us because this also shows that giving seminars is really helping, because there's one time I was giving a talk at a cardiology meeting, and basically presenting our sulfur knockout data, which can deplete pathological lipid level to near zero. And later, a clinician came to me and saying that the results are amazing, I've never seen before. And maybe this effect of such complete strong lipid lowering would allow the reversal of atherosclerotic plaques. And that's actually extremely or very significant in clinical settings. And now these results are interesting because now we have a much applicable, much controllable approach than a gene knockout. So, and this actually really intrigued or inspired us to perform a set of really tour de force experiment, because any of these single experiments actually took almost a year to complete. So what we did is actually to in neural models to first induce in a pathological phase to induce the formation of these atherosclerotic plaques as a result of elevated lipid levels. And then after this point, we start a therapeutic phase with this manganese-based regulation. And interestingly enough, we see a very nice reversal, and to some extent, very complete in some cases, of the existing atherosclerotic plaques in these live mice. And in some cases, the effect is so nice that we even need to break the y-axis to put some of these quantifications in the same panel. And certainly we are very excited about these, by the potential of this approach, which are completely originated from basic research, because we think that conceptually these are important because this shows that the atherosclerotic plaques, a major disease phenotype, can actually be effectively reversed, at least in immune models, and this practically can be achieved by intensive lipid lowering from by targeting really the condensation mechanism of this very essential, very general machinery of the COP2 complex with the manganese regulation we very serendipitously uncovered from basic study. But at the end, I should say that I'm not really promoting, I'm not really advertising that we should all eat or drink manganese, because I think what we know now about this element is interesting enough, but perhaps what we don't know about this is even more interesting, because in the field, it was considered that manganese is the least understood essential element. And so far, there's really no good sensors. There's, even in humans, there's no formal guideline for how much we should or we could take as humans, because it was all considered as a passive requireable for enzymes, and manganese deficiency is extremely rare. So we just need to take the basal amount. Now we're proposing that it has a completely different, very active signaling role. So the regulation actually will be very important, and certainly manganese, when inhaled, when inhaled through respiration that has neurotoxicity. However, a study have shown that dietary uptake, at least in mice, can go to very, very high doses, much higher than we used in our study. So really how to harness the bright side, but really avoid the toxicity or the dark side of this element, essential element, is certainly going to be interesting. And I think we are really move forward in this and really look forward to your advice. So at the very end, I would sort of summarize our journey so far by studying the cellular mechanism of the both the biosynthesis and the transport of these very important lipid-ferrin lipoproteins. And this, as briefly here, this is really a battle for us, but we hope to use at least two types of weapons. One, on the one hand, the very complex, very comprehensive human data, especially human genetics. On the other hand, very simple yet very robust virology. And I do hope that the combination of these two extremes in biology would help us to take our study, which was started from humans, eventually back to humans. At the end, I would really thank all the people who helped us in the studies that I presented today. And this is lab. I highlight people that who made important contributions during the talk and these funding resource. And in the end, I really thank you for all. Thank all of you for your attention. Be happy to take any questions if there's any. Yeah.
Okay. Thank you, Xiao-Wei, for the wonderful talk. Okay. So now the floor is open for the questions. I just now read the questions from the chat. The first question is from Professor Mao Yuxin, and he raised that do cell-4 deletion cells accumulate large amount of lipids or lipid droplets?
Oh, thank you, Yixing, for the question. Yeah, I always get asked for this. Yes and no. Cell-4 knockout do have some fatty liver, but not extremely severe. In the TMEM41B knockout, the fatty liver is actually, liver knockout, fatty liver is extremely severe. Cell-4 is actually much less, not even, not very severe. The difference is activation of lipogenesis by the very important regulator SREBPs. Basically, in cell-4 knockout, when you block in a later stage, the accumulated lipid would feedback inhibit SREBPs and therefore inhibit lipid production. So this would alleviate much of the lipid accumulation. And very interestingly, in our manganese treated mice, we not only do not see accumulation of lipids, we even see alleviation. No, we even see less lipid accumulation than control. So that part of the biology, we're still working on this. It was certainly going to be very interesting.
Okay, thanks, Xiao-Wei. So the second question is raised by Wu Shenjie. So he asked, does the magnesium affect other COP2 cargo secretion?
Yes, that's also a very interesting question. So specificity, right? We do see, so we did not exclusively look at screen a whole panel of secreted cargoes, but we do see a distinct selectivity between lipoprotein, which are affected by manganese, versus general secreted protein like albumin and many other plasma proteins. And so, but then the question becomes, where do these specifically come from? We think that it could be the lipoproteins are much bigger and they're very abundant. So maybe these type of secretion, either you need a lot of large vesicles, you need a lot of vesicles, or you need large vesicles, or you need a lot of large vesicles. And these are particularly sensitive to, requires really the very high efficiency of the COPII machinery. And these are the priority target for this manganese regulation. So in short answer, for albumin and a few other secreted cargoes that secreted proteins we look at, they are not affected by manganese. Yeah, and seems like lipoproteins are very unique in this sense.
Okay, thanks. So the third question is raised by Joshua Zimberg. So he asked, is there any indication of lipid regulation from the clinical chelator use, therapeutic or preventive?
Yes, this is very interesting. Yes, first of all, the chelators are very hard to use, especially we are using, so for us is intracellular metal signaling, and those are released actually from mitochondria. So to my knowledge, I don't think chelator may or may not work in this setting. However, we did obtain some human elements, even though these are association, these are marginal. So in a rural part of China, and there was actually manganese pollution in that area. So we were able to obtain several hundred samples from young adults, and those are very healthy. And we did observe a negative correlation, statistically significant, but does not account for a huge, not for a dominant effect. But we did observe that with higher manganese, those healthy adults appears to have lower plasma lipids. But these, I think, either clinical or human evidence, we need to look at this more seriously in a more calibrated, more extensive value. So yeah, certainly we're interested in that.
Okay, thanks. So another question is raised by Yue Xu. And this person asked, did you observe any lipid uptaking change by magnesium?
Yes, if I understand this question correctly, I can sort of reframe this. So would manganese affect lipid uptake from the small intestine? We did not specifically look at this, but my guess would be that manganese will also inhibit or decrease at least lipid uptake, lipid absorption from the intestine, which is very dependent on chylomicron formation and secretion. And would because the trafficking machinery, the trafficking program, we observed, we described on VLDL, it's very conserved, it's very similar to chylomicron. So I would think manganese would also inhibit chylomicron secretion, and that would decrease lipid uptake and absorption through the intestine. And that actually will also have some therapeutic implication. Yeah, thank you.
Okay, so the next question is raised by Tony Hui. He asked a general question. So is it a concern that targeting lipoprotein secretion cause accumulation of lipids in the liver, leading to fatty liver?
Yes, Tony, thank you for your question. I sort of touched upon this point earlier. Yes and no. And I did have a very thorough conversation with Henry Ginsberg, who is the guru of lipoprotein secretion. And he told me that in the field, they were looking for genes that decrease lipoprotein secretion, but do not cause severe fatty liver. And it seems like these would need to be at the distal part, at the later part of the of lipoprotein biology, especially at secretion. So the idea would be that you may have accumulation, but the accumulated ones would feed back and inhibit SREBP. I'm sure you are very familiar with that. And that would alleviate much of the fatty liver, and which would lower the risk of liver damage. And in our case, and I have to confess that we do not understand the mechanism yet, manganese treatment decreases plasma lipid, it also decreases hepatic lipids, at least in the disease models. So we do not understand the liver phenotype yet. It may dramatically decrease the lipid production by manganese. But if that's the case, that would be even more interesting to us. But we are we're trying to understand this. So a short answer to this is, seems like this idea of if you're hitting at the later stages of the secretion program, it may trigger negative feedback, and these would not lead to dramatic fatty liver.
Okay, thanks for the answer. So next question is raised by Feng Hua. So she asked, the why COP2 condensation has no effect on general secretion.
Yeah, and I think that the short answer may be the lipoproteins require a very high efficiency of COP2 assembly or vesicle production. This may have to do with the fact that the lipoproteins are both very big, and then they are also very abundant. So maybe that the lipoproteins need large vesicles and need a lot of them, or in both cases, need a lot of large vesicles. So they are particularly dependent on the maximal efficiency of COP2 function. And the general proteins may just get by with, normal COP2 function. And this is also true in the SOL1B knockout. Lipoproteins are affected big time in the SOL1B knockout, but the general secretion is not affected. This is it is also even true in chylomicron retention disease patients. Yeah, seems like there's a very distinct selectivity by condensation.
Yeah, maybe I just follow up on this question. Do you think that it has a specificity on the large cargo? These lipoproteins, collagens, or but other? Do you think? Yeah, just this is my question. Yeah.
I think it will be very interesting. I think we had the same conversation. It may even have some clinical implications because fibrosis have to do with collagen secretion. If you block collagen secretion, which is large cargo, if that's also sensitive to this condensation mechanism, you can over condensate COP2 machinery, inhibit collagen secretion. That actually will be very interesting. We should certainly look at this. We have not get around to look at this, but thank you for the suggestion.
Okay. Yeah, last question is raised by Chong. Oh, yeah, the question is always raised up. So the next question is raised by Chongyan Zhang. So he asked whether magnesium also condensation other factors, not only COP2, such as FAS. Yeah. Do you think that magnesium?
That's a fantastic question. I was exchanging the similar ideas with Steve McKnight. Either way will be interesting, but it seems in our hands that manganese regulation, fast condensation, first of all, is not affected by manganese. However, we can reconstitute this manganese dependent or manganese promotion of condensation by fusing a intrinsic disorganized element with a manganese binding peptide. So it seems like manganese binding may provide a seeding role to nucleate a few proteins to maybe lower the threshold of condensation concentration, and that seems to explain. That could well explain its promoting effect of condensation, but that would also provide a specificity because it requires manganese binding specific binding to as a requirement, as a prerequisite.
Yeah. So yeah, so many questions. So there is two questions remain. The next question is made by Wen Dai. So he asked, what is the trigger of magnesium release from mitochondria into cytosol, and could the cytosolic magnesium be taken up into the endoplasmic reticulum? Yeah, that's also a good question. What is the trigger to trigger the release?
Yeah, thank you. And this is something that we really would like to be helped, because the cellular dynamics of magnesium is not well understood. In and of the cells, there are transporters known, but within the cells, within organelles, it's very little known, as far as I can tell, not very understood. It is known that the uptake of manganese into the mitochondria is voltage dependent. So membrane potential is very important for this. In the case of viral, adenoviral triggered release of manganese from mitochondria, we think that it's because adenovirus kill the membrane potential of mitochondria, so manganese got leaked out. However, the transporters for manganese on the mitochondria is not well understood. We actually have evidence, and this was known previously. MCU can transport both calcium and manganese, and we now have some human genetic data also suggesting that this regulation is important for lipid regulation in humans. So we hope to wrap up the study very soon. But thank you, and this is really key. And we would love to get help in this area.
Okay, so last question. So, and yeah, he asked the person asked whether this magnesium has a specific effect on isoforms of Sec24. You all look at Sec24A, right? How about B, C, D?
Yeah, very interesting question. So short answer is affect all of the Sec24s, but we did not look at this extensively in a quantitative measure. And in fact, we have not been, I need to confess that, we have not been able to map the magnesium binding site because this may be a sort of, in the field may think that these metal ions could be a general post-translational modification, so may not have discrete binding pocket. But nevertheless, I think either evolutionary or compare the parallels would help us locate the residues that bind to Sec24. So yes, indeed, it affects all of the Sec24s as we have seen. But whether there's any quantitative difference or whether there's any specificity, very fine-tuned specificity, we don't know. And I think this will be very interesting to look at.
Okay, thanks. Thanks, Xiao-Wei. So for the very wonderful talk and also all address the questions so nicely. So, and I think yeah, it's a very nice. So Yihong, do you have anything to add on to this first talk?
No, I think the time is up, even though I still have a lot of questions, but I think we can find